In a muscle contraction, the brain sends a message to contract down the spinal cord to a motor neuron. Now, it splits into many different directions, but eventually ends up at the axon terminal. Now, once we get to the axon terminal, right up in here, we can see one of the many vesicles that is in the axon terminal. Now, it slowly begins to move towards the synaptic cleft. And once it eventually gets there, it releases its contents, which, among other things, include acetylcholine, or ACH. Now, the ACH bonds right here and opens this pathway to allow many of the other molecules to go through and enter the sarcoplasm. It also generates a muscle impulse which travels in all directions over the sarcolemma, and the T-tubules here allow the impulse to pass deep into the muscle to the sarcoplasmic reticulum, which releases its calcium ions. These travel throughout the sarcoplasm and eventually attach to the sarcomere. Once here, the calcium ions bond to the troponin, which causes it to pull on the tropomyosin, which in turn exposes the binding sites on the actin myofilaments. Now this allows the myosin heads to pull the exposed binding sites on the actin, which form cross bridges, and this ultimately causes the muscle to contract. The ATP then binds to the myosin and then splits into ADP and phosphate, which allows the process to repeat itself. The muscle begins to relax when the enzyme acetylchlorine esterase breaks down the acetylchlorine. The calcium ions are then transported back to the sarcoplasmic reticulum. As the calcium is brought back to the sarcoplasmic reticulum, a calcium troponin complex begins to be taken apart. Now, the troponin and the tropomyosin block the binding site on the actin, so the muscle fiber relaxes, and the ATP puts the myosin back in its original spot. Now the muscle is ready to contract again.